the most deformed animals from consanguinity, when incest destroys entire species. This dark history, known by few, has shaped the fate of entire bloodlines within the silent, isolated farms of rural America. For decades, consanguineous breeding practices have driven magnificent species to the brink of functional extinction, leaving them with such severe anomalies they defy our understanding of nature. Our investigation opens in 1953, within the remote mountains of Western Virginia. Here, the Peterson family operated one of the largest Appaloosa horse breeding properties in the United States. Harold Peterson, the family patriarch and a man of limited vision, firmly believed he could improve the breed through what he termed blood purification, the systematic crossing of closely related animals to fix desirable traits. At that time, understanding of genetics was extremely limited among rural breeders, explains Dr. Leonard Mitchell, a veterinary medicine historian at Cornell. Many believed crossing close relatives would intensify the animal's best qualities, without grasping the devastating biological consequences. More than 30 kilometers from the nearest town, the Peterson property sat in a valley surrounded by densely wooded mountains. This geographic isolation gave Harold the perfect environment for his experiments, free from supervision or interference. Few visitors were permitted, and veterinarians were rarely summoned, even when needed. In the early years, the results seemed promising. Appaloosas from the Peterson farm displayed stunning coat patterns, unique spots and markings that attracted buyers willing to pay high prices. But behind this apparent aesthetic perfection, a silent tragedy was beginning. By 1958, the first foals with visible anomalies were born. Atrophied front legs, abnormally small eyes, and mandibular deformities were just the start. Harold, however, dismissed these as isolated birth accidents and continued his practice, now crossing animals that were already products of consanguineous pairings. What we see in the surviving veterinary records from that period is alarming, says Dr. Elaine Harris, a veterinary geneticist. Notes from the only vet who visited in 1960 describe foals with immune systems so compromised they succumbed to common infections within days. Some were born with exposed organs or severe heart defects that prevented survival beyond a few hours. Between 1960 and 1963, the situation deteriorated rapidly. Adult horses began displaying abnormal behaviors, some grew inexplicably aggressive, while others developed repetitive motions, shaking their heads incessantly or walking in circles for hours. Mares had increasing difficulty conceiving, and when they did, they often suffered miscarriages or delivered stillborn foals. The case of the Peterson horses might have remained just another sad, undocumented episode in American animal breeding, if not for Raymond Foster. This young National Geographic photographer visited the region in 1964 for a project on the Appalachian Mountains. Foster arrived at the property by accident when his Jeep broke down nearby, says John Harrison, a local historian and author of Secrets of Rural Breeding, The Untold Stories. What he witnessed would forever change how we understand the effects of consanguinity on animal populations. The photographs Foster took, though never published by the magazine due to their disturbing nature, documented the result of 11 years of systematic inbreeding. They showed horses with deformed skulls, misaligned jaws that prevented proper feeding, atrophied limbs, and patches of hairless skin. Most striking were the eyes. Many animals had microphthalmia, incompletely developed eyes, or anophthalmia, a complete absence of eyes. What Foster documented was the complete collapse of a genetic lineage, states Dr. Paul Whitman, a geneticist at the National Institute of Animal Health. Records indicate that when Foster visited, roughly 78% of adult animals had some visible deformity, and the foal mortality rate had reached nearly 90%. Harold Peterson, however, seemed incapable of recognizing his own responsibility. In personal diaries discovered after his death, he blamed external forces, from water quality to alleged radiation from Western nuclear tests. He never considered his breeding practices were causing the disaster. Make sure you're subscribed to this channel so you don't miss any of our stories revealing the darkest secrets of human intervention in nature. These are truths few have the courage to face, but we all need to know them to avoid repeating the devastating mistakes of the past. When Virginia state veterinary authorities finally intervened on the Peterson property in March 1965, they found a situation even graver than Foster's photographs suggested. Of the 143 horses originally recorded, only 67 remained alive. Of those, just 12 met basic animal health standards. The official report contained descriptions that shocked even the most experienced veterinarians, explains Dr. Margaret Wilson, who accessed the state archives during her research for the Agriculture Department. There were horses with duplicated organs, incomplete digestive systems, and skeletal deformities so severe the animals could barely stand. One iconic case was Midnight Star, a 4-year-old stallion who had been Harold Peterson's pride. The animal, the product of 5 generations of inbreeding, had a black coat with perfectly symmetrical white spots, a trait Peterson had selected for intensively. Yet Midnight Star suffered from a rare cardiac condition, a heart with only 3 functional chambers instead of 4, leaving the horse in a constant state of hypoxia and fatigue. The case of Midnight Star perfectly exemplifies the fundamental problem of intensive consanguinity, says Dr. Thomas Reynolds, a veterinary geneticist. Harold selected only for the visible coat characteristic, without understanding he was simultaneously concentrating deleterious recessive genes that would normally remain hidden in a diverse gene pool. The science behind this disaster is clear. When closely related animals reproduce, the probability that offspring inherit 2 copies of the same harmful gene increases dramatically. In healthy populations, these harmful genes are diluted by genetic diversity, explains Dr. Harris. Think of it as a deck of cards. If you have a full deck, the chance of drawing a specific card is small. But if you remove most cards, leaving only a few suits, your chance of repeatedly drawing the same card increases exponentially. That's what happens with problematic genes in inbred populations. Lab tests on the surviving horses revealed alarming levels of homozygosity, where an organism has identical gene copies on both chromosomes. While healthy horse populations typically show homozygosity rates of 3 to 10%, the Peterson animals ranged from 42% to 68%. With such levels, it wasn't a matter of if genetic problems would appear, but how many and how severe, says Dr. Whitman. The immune system, embryonic development, metabolism, all fundamental biological systems were compromised. The effects extended beyond physical deformities. Veterinarians noted profoundly disturbed behaviors, mares systematically rejected their foals, and animals exhibited self-harm, biting their own flanks or repeatedly banging their heads against stall walls. We were observing the collapse not only of the physical body, but of neurological systems, explains Dr. James Bradford, a neuroscientist who later studied preserved brain tissue from the Peterson horses. Post-mortem analyses revealed significant malformations in the hippocampus and other brain structures crucial for instinct and emotional regulation. One of the most tragic aspects was the prolonged suffering imposed on the animals. Harold, unable to accept his practices were the cause, refused to euthanize even the most severely affected horses. Many lived for years, incapable of performing basic natural behaviors. The Peterson case was not just a genetic disaster, but a monumental ethical failure, states Dr. Elizabeth Morgan, a veterinary ethicist. By prioritizing a distorted vision of purity over animal welfare, Harold created a living hell for these creatures. Investigations revealed Peterson had falsified pedigree records, selling inbred foals as coming from diverse lines. Between 1955 and 1963, he sold roughly 200 horses to buyers in at least 12 states, spreading compromised genes through a significant portion of the national Appaloosa gene pool. The impact extended far beyond that isolated farm, says Dr. Mitchell. For decades afterward, Appaloosa breeders nationwide kept discovering genetic anomalies traceable to Peterson's animals. Some mutations persist in the breed to this day. While the Peterson tragedy unfolded in Virginia, an equally alarming, though less documented, situation was occurring in Clearwater County, Idaho. There, an isolated community breeding hunting dogs perpetuated practices with devastating consequences. The story of the Clearwater dogs remained obscure until 1971, when recently graduated veterinarian Dr. Sarah Jenkins took her first job in the region. What she found would transform her career and our understanding of inbreeding effects in domestic canids, says Michael Stevens, author of Lost in the Forests, The Forgotten Communities of the American Northwest. The Clearwater community, established in the 1920s, depended on specialized dogs for hunting. Over decades, they developed the Clearwater Hound, reputed to have superior tracking skills. By the 1940s, the local dog population had become extremely endogamous, with new genes rarely introduced. A notebook from Elmer Jenkins, a respected local breeder, revealed a detailed system favoring breeding between siblings, half-siblings, and cousins. When Dr. Sarah Jenkins arrived, she found a canine population in advanced genetic collapse. Her journal entry noted, "Clearwater hunting dogs display the most alarming set of congenital anomalies I have ever witnessed. Approximately 60% of pups were born with cleft palates and nearly 80% developed severe hip dysplasia before age 2. Most disturbing was Clearwater cataract, a condition where eyes became progressively covered by an opaque membrane, leading to complete blindness by age 4. Virtually all 287 dogs in the community could trace their lineage back to just 6 founding animals from the 1930s. The inbreeding coefficient reached 43% to 51%, astronomical levels, given that anything above 10% is considered concerning in canines. With each generation, conditions worsened. Dr. Jenkins documented litters where over half the pups had cryptorchidism, undescended testicles, spinal deformities, or severe heart defects. Behavioral issues were also rampant: compulsive tail chasing, self-licking to the point of injury, sudden aggression, and mothers rejecting or attacking their own pups. If you're watching and share my sense of outrage, leave a comment below. Nature deserves respect, not reckless interference. We need to amplify this message so such stories are not repeated. Despite the evidence, the community initially resisted Dr. Jenkins's recommendations. Elmer Jenkins argued that diluting them in blood would destroy the dog's special hunting abilities. "What we see is a recurring phenomenon," says Dr. Morgan. "Breeders often develop almost mystical beliefs about lineage purity, interpreting even deficiencies as proof of exceptionality." A tipping point came in 1973, when a severely affected litter was born. Of 9 pups, 7 had partial anencephaly, incomplete brain and skull development, and the 2 survivors displayed such dangerous behaviors they had to be euthanized before 6 months. This finally broke the community's resistance. With reluctant permission, Dr. Jenkins implemented a genetic rehabilitation program, gradually introducing healthy outside stock while maintaining meticulous kinship records to prevent new inbreeding. Her program represents one of the first documented genetic rescue efforts in a canine population, says Dr. Thompson. Her notes provided a model used in similar cases worldwide. The recovery was long and methodical. Dr. Jenkins's strategy had 3 pillars: controlled introduction of new breeding stock, genetic mapping to avoid inbreeding, and rigorous monitoring of physical and behavioral traits. Without modern genetic sequencing, she created a diversity scoring system using clinical observation and records alone, says Dr. Catherine Miller, a conservation geneticist at the University of Colorado. She was practicing advanced population genetics with the tools she had. Over the first 2 years, 14 outbred dogs were carefully introduced. Initial results were encouraging. By the 3rd generation, cleft palate incidents had fallen from 60% to about 12%. Behavioral problems proved more stubborn, often lingering for generations. A severe snowstorm in winter 1976 tested the community's commitment when 2 key outside breeders died. Instead of reverting to old practices, community members traveled through the snow to secure new, unrelated dogs. Dr. Jenkins noted in her journal, the community met to cover the costs. "The old ways are dying with us," one said, "but the new ones will live with our children." By 1980, results were undeniable. The canine population had grown to about 350 animals, with average inbreeding coefficients reduced to 8.3%, down from 47%. Functionality had not only been preserved but improved. The healthier dogs proved superior hunters. Corneal dystrophy, once nearly universal, now affected less than 5% of dogs. Severe skeletal issues and abnormal behaviors had dropped dramatically. Dr. Jenkins's work was published in 1982, bringing international attention. The most important legacy was the practical demonstration that severely compromised populations can be recovered with methodical intervention, concludes Dr. Miller. The principles she established have been applied to everything from workbird breeds to wild populations in fragmented reserves. Yet this scientific legacy does not erase the suffering. As Dr. Jenkins reflected, every documented malformation, every observed abnormal behavior represents a sentient life experiencing unnecessary pain. True triumph will come when such interventions are unnecessary, when we finally understand that genetic diversity is not just statistically desirable, but ethically imperative. While the Peterson and Clearwater cases may seem like relics, similar practices persist today, often masked by claims of preservation or breed purity. The phenomenon is more insidious now, explains Dr. Elena Rodriguez, an expert in animal welfare and breeding ethics. One emblematic modern case is the English Bulldog, a breed with a gene pool so restricted that genomic studies suggest its current diversity is equivalent to that founded by only 68 unrelated dogs. The modern Bulldog is essentially a walking warning about inbreeding dangers, says Dr. Whitman. Most suffer severe brachycephaly, causing chronic breathing problems, and over 80% require C-sections because natural birth is impossible. In dairy cattle, 99% of U.S. Holstein bulls used in artificial insemination can trace their lineage to just two 1960s founding animals. We've created cows that are efficient short-term milk machines, but fundamentally dysfunctional as biological organisms, says Dr. Frank Anderson. These cows typically survive only 4 to 5 productive years, versus a natural 15 to 20-year lifespan, and face severe fertility issues. In show Arabian horses, selection for an extremely concave dish face has led to widespread neurological, respiratory, and dental problems. Some award-winning horses have cranial malformations that would have been considered severe deformities 50 years ago, notes Dr. Wilson. A conspiracy of silence often surrounds these issues in the show world, says Dr. Howard Bennett, a former dog show judge turned critic. Behind the scenes, everyone knows. But publicly, we keep rewarding the most extreme specimens, perpetuating the cycle. For wild species in conservation programs, the lessons of Peterson and Clearwater are critical. The cheetah, for instance, has passed through severe genetic bottlenecks. Modern cheetahs are so genetically uniform they are practically immunological clones. This means a single disease could decimate the entire population, explains conservation biologist Dr. Samantha Lewis. This homogeneity manifests in poor sperm quality, small litters, and high cub mortality. We know better, but we often don't do better, says Dr. Rodriguez. Historical cases should serve as powerful warnings, but instead we repeat the same errors, now amplified by advanced reproductive technologies. There are, however, signs of change. In 2019, the UK Kennel Club implemented new guidelines for brachycephalic breeds. Outcrossing programs, like the one for Dalmatians that introduced a pointer cross, show genetic problems can be addressed without losing defining traits. In conservation, programs like the one for the Mexican wolf use genomic analysis to maximize genetic diversity in captive breeding. We are finally recognizing that preserving numbers alone isn't enough, says Dr. Lewis. We must preserve functional genetic diversity for long-term survival. A numerical success that results in a genetically compromised population is not true conservation. As we advance in the 21st century, the lessons of Peterson and Clearwater remain vital. They remind us that our power to shape other species carries an obligation to respect the biological principles underlying health and welfare. The true legacy of these cases lies in the lessons they offer, concludes Dr. Rodriguez. Respect for biological integrity and genetic diversity is not just scientifically sound, it is ethically imperative. If you've made it this far, you're invested in this story. Leave a comment saying "watched here" so I can get to know who truly appreciates these channel stories. Be sure to subscribe to follow our investigations that reveal the darkest and most educational truths about human impact on the animal world. Leave a like to support this work of awareness, and share with friends and family who need to know these important histories about the consequences of animal consanguinity. Together, we can promote a future where respect for genetic diversity and animal welfare guides our breeding and conservation practices.